grandpa, who does it? Then he said, what grandpa can tell? He said, beta, there is a bad wolf in the world, and this bad wolf, no, he is doing all these bad things. हो गया start? Okay. Record भी शुरू किया? Okay. So maybe I'll come to that incidents later. Let us first, because we have to finish in time. Yeah. So maybe we'll start again. Yeah. Okay. So shall we finish? Yeah. So then boy said, I mean, who is doing all these bad things? And the grandpa say, there is a bad wolf. Who does it? And then he said, boy said, where does that bad wolf stay? Where does he reside? And he said, no, like there is a, I mean, there is a place in a heart of each and every person, no, in this world, and there this bad wolf stay. So, and then he said, he added, beta, there is good wolf as well. So that also stays in the same place. And he says, ki always there is battle going on between this bad wolf and good wolf. And then he says, finally, only one wolf wins. So he asked the question to boy, what do you think, who wins? Okay, so same question if I ask you, what do you think, who wins? Which wolf wins? What will you say?
To whom you give power.
The one that we want to win. Okay, he said the one whom we give power. So how we give power to that particular wolf? कैसे दे सकते हैं? By doing the things that, yes. So to be more specific, the wolf whom we feed more, he will win more. That particularly, that will win. So how to feed good wolf and how to feed bad wolf? Feeding bad wolf, very easy. You must have seen in nowadays whatever happening around you, you type some words in your mobile, you start feeding the bad wolf, right? Everything is in the tip of your fingers. You look around, whatever happening in the society, all bad drugs and drinking and smoking, you feed bad wolf, right? So how to feed good wolf? Avoiding all these things, being in a good company, having good friendships, right? Reading good books, isn't it? There हम पे जो संस्कार है बचपन से, है ना, जो मम्मी पापा ने बोले है सारी अच्छी चीजें, डूइंग ऑल दोज थिंग्स हियर एज वेल. यहाँ पे ऐसे नहीं, अभी कौन देख रहा है हमें, मम्मा पापा थोड़ी ना मॉनिटर कर रहे है ना. ये छोड़ के कंटिन्यूइंग ऑल दोज गुड थिंग्स इज लाइक फीडिंग द गुड वुल्फ. व्हाई आई टोल्ड यू, बिकॉज सी यू आर इन अ scenario now, first time you are out of the home, away from all bondages, feeling free, and that is the time when there is a like chance to slip, and that's why you have to cling on to all those good things. Let us come to our lecture today. Technical glitch is over. Yeah, so last histology lecture you were taught about connective tissue, right? So, and you must have been learnt that connective tissue can be divided or into two types. What are they? Loose, so more like generalized division, general connective tissue and special connective tissue or modified connective tissue, right? And then we have seen last time what are the general connective tissue. Okay, now that time you must have been told that which tissues are called modified or special connective tissues. Any idea? Yeah? Bones, cartilage, blood, right? Examples of modified connective tissue. So in the days to come we are going to learn in histology about this modified connective tissue, and today one of that, the bone, histology of bone we are going to learn. So what do you know about bone? It's already taught in general anatomy. You already learnt in maybe your 7th, 8th, 9th grade somewhere that time. So what is bone? What do you understand by bone? Yes, yes beta. Okay, It consists of different type of cells, fibers, matrix. Yeah, right. And framework बनाता है body का, it's a function, right? And locomotion, movement में it helps us. Yes, exactly right. Yeah. So all of you are correct, right? Now, as we are discussing histology, let us have a look on the histological aspects, how we define it on a histological point of view. So, it is a specialized type of connective tissue, right? So, bone is what? It is a specialized or modified type of connective tissue, and it has extracellular matrix containing calcium salts. That is the more specific point about this. Okay, it's having calcium salts as in a matrix. So, as a connective tissue, as he mentioned very correctly, it consists of cells and the matrix. Okay, matrix consists of ground substance and the fibers, isn't it? Now, this materialized extracellular matrix which is present there, no, as mineralized actually, this mineralized matrix, it gives the property of bone which is hardness, right? So, it is because of this mineralized matrix. And what is that mineral? Calcium salts, right? So, and yes, it is a living tissue, and it gives response, it shows changes in response to the physical stress and hormonal changes as well. Okay. So, that is all just introductory points about bone, and as you have mentioned very correctly that it gives support, it helps in locomotion, isn't it? It is having protective function when we consider thorax, when we consider brain, right? These bones are having protective functions, and along with that they also act as a storehouse of this calcium and the phosphates. And we should not forget about this function. What is it? Read loudly. Hematopoietic function. What do you understand by that? Production of different type of cells, right? And where does it happens actually? Bone marrow. Yeah, very good. Now, when we try to learn some features of the bone, no? So, we have already learned its classification in general anatomy, right? We are divided into like long bones, short bones, irregular bones, flat bones, right? Different types. So, now when we take a section of the bone, and we can easily see that there is a epiphysis, the two ends of the long bone, right? We call them epiphysis, where growth is continuously taking place, and middle part is the shaft. And when we take a section at the epiphysis, we can see there's many cavities, small cavities. That particular part is filled with cavities, and that's why it looks like a sponge, and that's why it's called spongy bone. And in that spongy bone we can see there is a bone marrow, and also like in the middle part there is a cavity that is called medullary cavity. There is also we can see the bone marrow. So, we will not go in detail about what type of bone marrows and all. Okay. So basically we know long bone have epiphysis and diaphysis, right? And then like histologically, if we see, we are going to see detail in the coming slides. We can divide the bone into compact bone and the spongy bone, as you can see in this diagram as well as in this diagram. So this peripheral part which is dense, so where we cannot see any kind of cavities, it's a dense continuous structure. This is the compact bone, and here other part where we can see many cavities, this is the spongy bone. Same thing. So as this is like long bone, but if you see the flat bone, here also like if skull bones are not yet taught, but just I think when we discuss the different types of bones, that time skull bones were given as an example, right? So where there we can see there are two tables, like there is an outer table, there is an inner table. So outer table and inner table, they are compact bone, and in between them there is a spongy bone. So this is how we can divide them, and these bones are covered by the coverings. There is on the outer side there is periosteum, on the inner side there is endosteum. So this endosteum and periosteum, again detail about this too we are going to see, and there are two type of marrow, red and yellow. So yeah, this is clear. Now let us come to the coverings of the bone. As you can see in this diagram, okay. So all these diagrams are taken from the textbooks which we have recommended you to use. Okay, we have put the list there. So and so you can see even the names of the authors. So this is the, what is this? Can you read? Which covering is this? What do you think? This has to be periosteum on the outer side, and this is towards the medullary cavity. So here there is a endosteum. So two coverings there towards the medullary cavity, the inner aspect, that is endosteum, and towards the outer aspect, that is periosteum. So when we say, see periosteum, no, it is like it is on the external surface, and it has it is covering the bone everywhere except where there is a muscle coming and attaching the bone. At that point periosteum won't be there, and when the bone is covered by articular cartilage forming the joint surface, that at that place also periosteum will not be present. But at all other parts of the bone is covered by periosteum. And again this periosteum is having outer fibrous layer, as you can see here. This is the outer fibrous layer, and then there is inner cellular layer. These two layers, inner cellular layer is a osteogenic layer, so which contribute to the other cells, bone forming cells or other cells in the bone. So outer fibrous layer and inner cellular layer, these are the two layers of periosteum. Clear? Then there is an endosteum, as you can see. Is it visible in this diagram? So this endosteum again is a membranous layer and covers the inner surface of the bone, as I mentioned already, towards the medullary cavity part of the bone. And then as compared to the periosteum, you can see here, this is endosteum, which is thinner. So endosteum is thinner as compared to the periosteum, and it is having only one layer, single layer. Periosteum we have seen outer fibrous layer, and then the cellular layer also made up of 2-3 layers of the cells, but here endosteum, here we can see only single layer of the cells, and most of the cells are the cuboidal in shape. So that is endosteum. So we as we already discussed, bone is a living tissue, it is a dynamic tissue. Okay, it shows response to the stress, any kind of injury, it shows response to the hormonal changes as well. Now in the living, but what happens after death? After death we can now we are using these bones of those individuals who have donated their bodies, right? We have recovered them, and now we are using them for the classroom teaching. So we dry them and use them for classroom teaching. Now what happens? No, these, when we dry this dry bone, are not actually living bones, so we cannot find in them the periosteum and the endosteum. These are not there. There are no blood vessels, there are no bone, no nerves in the dry bones. One thing we have to understand is the sharpest fibers. So sharpest fibers, these are the collagen fibers basically, and what they do is, as you can see here, see this is the periosteum, here is the part of the bone, and these sharpest fiber are helping the periosteum to get connected, get attached to the bone proper. So these are the collagen fibers. Sharpest fibers are the collagen fibers which bind periosteum to the bone. Is it clear? You are with me? Yes, yeah. They are also called as bone fibers or the perforating fibers. And we have to remember that there is a, like, what is osteon we are going to see in the coming slides. So functional unit of the bone. So they do not enter in that part in the osteon. They just lie out on the outer aspect, and they are continuous with the extracellular matrix of the bone. So these are the points you should know about the sharpest fibers. So basically these are the fibers which are connecting the periosteum to the bone proper. Now when we say composition of the bone, what are the elements, what are the making the bone? Okay, so bone consists of basic 3 components. So as we have discussed very early when we are discussing about the introductory part. So bone is having cells, bone having fibers, and bone is having ground substance. So, one by one about all these three components we are going to learn today. Yeah. Now, how we can classify the bone? So, depending on like we have seen that when we see, we take a bone, cut it, take a section of it and observe it with the naked eye. What we can see is, we can see compact bone. We have already discussed the outer part which is not having cavities is the compact bone, and then we can see there is a cavity which is the bone which is having so many cavities, that is the spongy bones, right. So, this is how we can classify compact bone and the spongy bone. Then another way of classification is depending on their histological appearance. Okay. So, now there is a immature bone, it's or woven bone. So, during embryonic life, when bone formation is taking place, that time bone is called woven bone. So, even in the adult life, when after fracture, when healing take place, okay, when bone formation, new bone formation is taking place, that time also we can see the woven bone. So, basically woven bone is an immature bone, it is also called as primary bone. So, if there is a primary bone, there has to be secondary bone, or if there is an immature one, there has to be mature bone. So, mature bone is called lamellar bone. So, what happens now? Actually, the adult bone is having very different lamellas. So, the structure of the lamellas we are going to study in the next slide. Okay. So, lamellar bone, these are the secondary bones, and they are having lamellar pattern of the arrangements of the components. We are studied three components are there, like. So, these components will be arranged in patterns of the lamellae, and that is why that particular type of bone is called lamellar bone. Hope it is clear. Yeah. Now, here we can see that lamellae. So, basically lamellae are the thin plates, okay, which are kept one above the other, and then these, what basically lamella is, so it is parallelly arranged type 1 collagen fibers ka bundles. There are so many collagen fibers coming together forming the bundle one above the other, and many such bundles together form one lamella, and many such lamellae are arranged one above the other, okay, giving rise to lamellar pattern of the bone. So, these lamellae, as we have discussed, as we can see here, they are arranged one above the other, and now when they are arranged one above the other, some small spaces are created, some small spaces are there in between these lamellae, and these small spaces we can see here. So, these are called as lacunae. What they are called as? Lacunae. Is this clear so far? So, what we have seen is, so lamellar bone, basically the adult bones are the lamellar bones. We call them secondary bones as well. So, they are made up of lamellae. Now, what is lamellae? What is lamella? Parallelly arranged bundles of collagen fiber. There are many collagen fibers forming a bundle, many such bundles running parallelly, and they form lamella. Many such lamella are put one above the other, right? And when they are put one above the other, some spaces are formed in between these lamellae, and these spaces are called as lacunae, right? Now, even you can see here, so when these collagen fibers, when we say in the lamellae, these collagen fibers, they are parallel to each other, like this is a lamella, above that is another one, above that is another lamella. Now, collagen fibers in these lamellae, they are not like in one lamella, they are parallel, they are parallelly running bundles, but the other adjacent lamella, they are not parallel, they are in right angle, perpendicular to the adjacent lamellae. Is it clear? So, this is the arrangement of the collagen fiber bundles in individual lamellae. Now, we have talked about lacuna, right? What is lacunae or lacuna? Space between the two lamellae, right? Now, what its significance is, this lacuna contains osteocyte. Osteocyte is a cell of bone. There are different cells in the bone, we will see that. So, one of that is osteocyte. So, osteocyte is present in that space called lacuna. Now, this lacuna, so this is a lacuna. Can you appreciate this dark black space is a lacuna, Okay. In the lacuna, can you appreciate this white one is the osteocyte. Now this lacuna is connected to the adjacent lacunae by channels called canaliculi. So this is one lacuna, this is another lacuna, and this is the channel. This is the channel. This is the channel connecting each other, the lacunae to the each other. These are called as, say it loudly, canaliculi, right? So what is present in the lacula? Osteocyte. What is present in the canaliculi? Cytoplasmic extensions coming from the osteocyte. Is it clear? Yeah. So what happens now? Now this lamellar pattern or this canaliculi which are there, I mean there are lacunae, there are canaliculi, lacunae occupied by the osteocytes and canaliculi by their cytoplasmic processes. So whatever the stability or density to the bone is provided by, I mean these cells contribute, osteocytes and their processes contribute in their strength, or we can say their stability, along with the matrix which is there. So these lamellar bony arrangements, so they are there in the compact bone as well as we can see them that in the spongy bone as well. Is it clear? Yeah. Now these lamellar bones replace the woven bone. So initially when bone formation is taking place, what type of bone is there? There is a woven bone or immature bone that is replaced by this lamellar bone. Okay. So let us have a look on the woven or immature bone. So basically what happens now, it is like, we can also call them fibrous bone, and it show organized arrangement of, there are collagen fibers, and these fibers run in different direction. So collagen fibers are not, what happened in the lamellar pattern, they are in a bundles, isn't it? They are forming dense bundle together, running parallelly, and okay, and forming a lamella. But what happens in woven bones, these collagen fibers are running haphazardly in different directions. That is the main difference between the woven bone and the lamellar bone. And yes, when there is any new tissue is formed, lots of blood vessels will be there, isn't it? So we can see those blood vessels when this woven bone is getting formed, or immature, or the primary bone is being formed. Now what happens in, there is a disease called Paget's disease. In what happens in this is, like, normally woven bone or the immature bone shall get replaced by the lamellar bone, isn't it? When this does not happen, when there is a non-conversion of this newly formed woven bone into lamellar bone, then that particular disease is called Paget's disease. It was discovered by the scientist called this Paget's. So James Paget, and after his name, it is this disease is given the name Paget's disease. Okay. So what will happen to the individual? What do you think? Bending. Yes, there will be no hardness in the bone. That's why there will be bending, isn't it? Bending of the bones. So where the such individuals will be having bended, like the bending in the feet or legs or their upper limbs everywhere, right? So now let us come to the compact bone. So this compact bone do not show the cavities, as this here we can see the cavities, spongy bone, but if you have a look on the compact bone, there are no cavities. So basically there are no cavities on the gross examination. Then basically where we find them, in the diaphysis, okay, of the long bone, and even outer table, inner table of the flat bones. So where there we can locate them. So here their outer surface is covered by periosteum and inner surface covered by endosteum. We have already seen that outer covering and inner covering. And then when we take a section of this ground bone, no, actually when we are going to study in a histology lab today, in our practical today, we are not going to see the endosteum and periosteum, because it's a dried dead bone, isn't it? We acquired it from a dead individual. So periosteum and endosteum won't be visible in that. Now what we can see is lamellae, and these lamellae arranged in different pattern. So now we are going to see these lamellae in three type of patterns: Haversian system of lamellae, interstitial lamellae, and circumferential lamellae. These three type of patterns we are going to see. Okay, when we examine the bone tissue in the histological lab under microscope. So, what we are going to see in the histology of compact bone, we are going to see osteon. So, osteon is what basically it's a functional unit of the bone where there is a canal in the center surrounded by lamellae, and there are Volkmann's canal and there is a Haversian canal. These two canals we are going to see in the coming slides, okay. And then along with that we are going to see interstitial lamellae and the circumferential lamellae. So, as you can see in this diagram, osteon. So, osteon, as I mentioned earlier also, it is a structural and functional unit of compact bone. So, what do you see here is this entire circular structure, this is osteon. Now, how osteon is formed? It is having a central canal. So, basically if you try to imagine three-dimensionally, imagine one pipe going straight and there are some structures surrounding it on the circumferentially. So, what is happening here is there is this Haversian canal, and this Haversian canal which are running parallel to each other, different Haversian canals, and they are surrounded by this lamellae and making this rounded structure. This is called osteon. So, now what happens when we are preparing this bone? No, basically these Haversian canals contain blood vessels, they contain nerve fibers as well, but in dry bone, when we are going to study them in histology, everything is lost. As I told you earlier only, when we are going to study, there will be no blood vessels, there will be no nerves, there will be no periosteum and endosteum, right. So, as there are whatever contents are there inside this Haversian canal, they are washed away, they are not there. So, basically what happens, no, they are filled by sometimes some little bit dust. That is why that sometimes we may see them in a black in color, sometimes there is no dust, we can see them white in color. So, that is the Haversian canal. Now, this Haversian canal, we can see they are surrounded by concentric lamellae, and then as you can see in this diagram, so this is one lamella, this is another lamella, this is innermost lamella, and in between the lamellae, can you see these spaces? What are these? Lacunae. These are the lacunae. And then can you see these processes? These are the, say it loudly, canaliculi, right. So, we see basically in section, in the histological section, what we are going to see is we are going to see Haversian canals surrounded by laminae, concentric laminae. In between the laminae, we are going to see the lacunae and the canaliculi, right. And now, as I already told you, canaliculi, what they contain? They contain cell cytoplasmic processes of the osteocytes, and lacuna contain the osteocytes proper. Yeah. Now, this vertically running Haversian canals are connected to each other by other channels. These are called as Volkmann canal. So, this is Volkmann's canal, this is Volkmann's canal, this is Volkmann's canal, okay, and they run perpendicular or oblique to the Haversian canal. And now, the point to remember is these Volkmann's canals are not surrounded by this concentric lamellae, Haversian canals are surrounded by the lamellae. Yes. So, what we see in the transverse section of the ground bone, we are going to see Haversian canal, they appear as oval or circular hole, as you can see here. We are going to see Volkmann canal sometime, not always, in all sections we are not going to see Volkmann's canal. If that particular section pass through that, then only we can see Volkmann's canal. And then, as I told you, Volkmann's canal is not surrounded by concentric lamellae, Haversian canal is surrounded by concentric lamellae. So, this is how we are going to see under microscope. So, what is this? Say it loudly. Haversian canal. So, these are the Haversian canal, this is Haversian canal, all this, okay, and here this Haversian canal connected to the other by this Volkmann's canal. So, as I told you, most of the places we will not, some places we will see this Volkmann's canal, okay, in the transverse section. So, basically this is transverse section. Now, what we see here, there is one lamella, other lamella, other lamella, there are concentric lamellae like surrounded by circumferentially, right? And between these lamellae there are spaces, the lacunae, and the lacunae are showing the processes or branches. These are canaliculi, right? So, and this one structure together, it is called as osteon. Osteon is a functional unit of the bone and functional and structural unit of a bone, right? And as you can see here, these are the lacunae and these are the processes. Yeah. So, this is the longitudinal section of the dry bone. So, what we see in the longitudinal section is this. What we see is this running, vertically running, these are Haversian canal, which are surrounded by, these are the lamellae, concentric lamellae, and then these two Haversian canals are connected by Volkmann's canal. Yeah? So, this is what we are going to see in the longitudinal section. As far as like practicals are concerned, we have two slides of the bone. One is of the dry bone ka transverse section, another one is dry bones ka vertical section. Okay. So, or longitudinal section. In the longitudinal section, we are going to see the Haversian canal running perpendicular or parallel to each other, running vertically. They are connected by oblique or horizontal Volkmann's canal, and we are going to see in between the lamellae. And in the transverse section, we are going to see the osteon, the functional unit. Okay. So, osteon are the circular structures, round circular structure, where in the center there is a Haversian canal, and that Haversian canal is surrounded by concentrically placed lamellae, that is the osteon. So, this is what we are going to see. Now, along with that, what we are also supposed to see is lamellae. So, now what happens now? May see outside, we can see there is external circumferential, or there are circumferential lamellae. So, which are on the circumference, which are on the outer side or towards the periphery. Okay. So, they are on the outer side, then we call external circumferential lamellae. Then when they are towards the inner aspect, inner circumferential lamellae. And now what happens now, there are some lamellae in between the osteon. See this one here. So, we call them as interstitial lamellae. So, we are going to see this interstitial lamellae, circumferential lamellae, and osteon, these three main structures in the transverse section of the dry bone. In the longitudinal section, what we are going to see? We are going to see vertically running which canal? Horizontal canal, which are connected to each other by oblique or horizontally placed Volkmann's canal. Yeah. Now, here just a schematic view to make you understand. Like here we can see this is one lamella, okay, another lamella, three lamellae placed one above the other, or concentric lamellae, and in between the space, that is the lacuna. Lacunae is occupied by this osteocyte, and osteocyte is showing this cytoplasmic processes which is present in the canaliculi. Hope you have understood this, right? So, when we look the slide under low microscope, low power, no, we are going to see this. So, we are going to see these osteons, okay, and we then we will be seeing interstitial lamellae, as well as, if you find, we can see outer and inner circumferential lamellae. Now, to label them is not, I mean, it's not possible when we get the slide here, because we don't know which is the outer, which is the inner surface. We can simply label them as circumferential lamellae and the interstitial lamellae. And this is high power view, or you can say 100x view of osteon proper. So, where here we can see here the Haversian canal, and they are surrounded by concentric lamellae. So, we can see lacunae here, and we can see this process canaliculi. And yes, in between them we can see the interstitial lamellae as well under high power. Now, here is the transverse section, longitudinal section, where we can see this Haversian canals, and these are the Volkmann's canals. As far as the slides which we have, there are some slides where we can see this Haversian canal very nicely, and there are other slides where these Haversian canals are broken in small pieces. So, we have to appreciate accordingly. When you see the slide, like we will be there in the lab, you call us and get your doubts cleared if you get any doubt when you see the slide. Yeah?
The previous one, the parallel lamellae of the next one,
Yeah. Here,
they are the same as the concentric only?
these lamellae, no?
The parallel lamellae.
Yeah. So basically, longitudinal section that they will be parallel to each other. In transverse section, they will be concentric. Yeah. Yes. So we have talked about, but let's have a little more about interstitial lamellae. So interstitial, what happens? No, when this bone is getting formed, no, so that time newly formed these osteons are there. So when older osteons are there, no, their lamellae, these lamellae are called interstitial lamellae. So between adjacent interstitial lamellae and lacunae, and so these all these lacunae and even radiating canaliculi also present. And circumferential lamellae, we have just talked about outer circumferential lamellae and inner circumferential lamellae. When this in this schematic diagram, we can label them because we know that here is the like towards the medullary cavity and this is the outer aspect. Okay, but usually when we get the tissue in our lab, that we cannot label it whether it is a external or internal or inner or outer circumferential lamella. Now why that? Because then I will explain how the slide is prepared, then you will understand. We have talked about periosteum already. It is having outer cellular layer and inner outer fibrous layer and inner cellular layer. Inner cellular layer is also called osteogenic layer, and this is we have to remember that it is highly vascular tissue. So these vessels and lymphatics which are present there, they communicate with the vessels of the Haversian canals and the Volkmann's canals. Endosteum, we have talked about the inner aspect. They are lined by single cell and they consist osteoblast, osteoclast, and they are flat, most of the time flat, or the low cuboidal in shape, these cells. So what we have seen so far in compact bone, we have seen like there are no cavities, okay, and we can see in the compact bone osteon, the functional and structural unit. We see interstitial lamellae, we see circumferential lamellae, and then we have seen in between the lamellae there are lacunae. Lacunae is the space for osteocytes, and then lacunae give have we can say canaliculi, and canaliculi are occupied by the cytoplasmic processes of the osteocytes. Yes. Yeah. Now, how we prepare the slide? In the earlier class, you must have been told about we take a small piece, isn't it? Take a sections of it. There is a special technique, some process goes on for that. Now, what happens now? There is an instrument called microtome, which cut any tissue into small slices. So, around 5 to 10 micron is the thinness, or you can say thickness of that particular tissue. Then we can observe it under microscope properly. Now, what happens now to make that sections is not possible because it's a very tough tissue. Bone is a tough tissue. So what we do is we make a small piece of it, maybe around 2 by 2 centimeters, and we hold it and we ground it. Ground it means हम उसको घिसते हैं. There is a bone, grounder bone. So we ground it so much long that it becomes very thin and transparent. And when it becomes so thin and it looks like a transparent structure, then we do further processing of it, like we put it in the alcohol, we put it in the xylene. There are steps of preparation the slide, and finally we can put it in the DPX mounted and we put it in the slide, put cover slip of it on it and observe it under microscope. Another thing is, as it is having many inorganic substances, right, mineralized salts, it does not take up the stains, and that's why we are going to see bone as a colorless slide. As you must have observed this diagram, no? No eosine and no hematoxylin stain, no pink and blue color. So that is what, so easy to identify the bone slides in the spotter, very easy, because only 2 slides where staining will not be there, isn't it? There will be one more slide will be there without stain. In the next class we are going to teach you. But bone tissue identification in the spotting is very, very easy. Okay. So easiest slide to identify is the slide of the bone, whether it is transverse section or the longitudinal section. So I told you about the preparation. Let us have a look on the spongy bone. So as we have said that like it's also called cancellous bone or trabecular bone, and like we can see that it looks like a sponge. It has a spongy appearance because there are many cavities, and because of that it is because of that we call it spongy. And when we say this particular bone, no, it do not have Haversian system, it's have trabecules. But as I told you earlier, there are lamellae, right? Now in this trabecule what we see is osteocytes, they lie in a cavity. There are also lacunae. So these osteocytes will be present in those lacunae, and then this radiating canaliculi also there, like as we have seen canaliculi in the compact bone. This will be connecting the other canaliculi, surrounding canaliculi. And these trabeculae are thinner, so there is no need of Haversian system there. So what we see in the spongy bone, we see trabecular network. So network of irregular bars basically, which osteocytes in the lacunae will be present. There will be canaliculi, like there are sponge like cavities. So we call it as a marrow cavity, and then there are no Haversian system, no Haversian canal, no Volkmann's canal, because the nutrition which is needed, that will be from the marrow cavity only. It can receive it. And when we take a observe it under microscope, when we prepare a slide, we can see like this. So this slide, no, spongy bone ka slide we do not have with us, but it will be looking like this. So what we see, we can see bony trabeculae like this. These are the trabeculae, and inside the trabeculae, can you appreciate these lamellae? There are the lamellae, then there will be lacunae, there will be osteocytes in the lacunae, and lacunae also they will be showing the processes canaliculi, right? All this will be there, and as like there are the spaces in between which will be having marrow. Marrow will be like producing the hematopoietic cells, right? So that's why we can see this hematopoietic tissue over here. So all these are the marrow cavities with the hematopoietic tissue, and we can see here the osteocytes which are present in the lacunae. So if we see it under high power, we can see like this. These are the bony trabeculae, and inside the trabeculae we can see this lamellae and the osteocytes. We can see the marrow cavities filled with the hematopoietic tissue. So here, if this is a high power, we can see here, like inside the lacuna there is the osteocyte. These are the osteocytes present in the lacunae. Yeah. So let us have a quick look on the other components. So we have talked about there are cells, there are organic components, so bone matrix and the cell. So cells, basically there are osteoprogenitor cells, or we can say stem cells, which are going to give rise to other type of cells. Then there are osteoblasts, then there are osteocytes, there are osteoclasts, okay, and there are other cells which are called bone lining cells. Matrix can further be subdivided into like organic and inorganic. What do you understand by organic and inorganic? Yes, beta? Yes? Organic is the living one, is a non-living, as simple, right? So basically organic component, we see type 1 collagen fibers, and we can see this proteoglycans and the glycoproteins. And inorganic, we see mainly the calcium phosphate. Main content is the calcium phosphate, along with calcium carbonate. There are some fluorides and magnesium and sodium. Okay, these are the components which are forming the inorganic. So together all these form a modified connective tissue. We call it as a bone, right? So now let us have a quick look on the cells. So osteoprogenitor cells, like these are derived from the mesenchymal cells. Mesenchyme, what do you understand by mesenchyme? Embryonic connective tissue, we call it mesenchyme. So embryonic connective tissue cells are mesenchymal cells. So from them these osteoprogenitor cells are derived, and what they do, they give rise to osteoblasts. Now next cells are the osteoblasts. Here we can see these are the osteoblasts. These are the bone forming cells basically. So what osteoblasts, they form single layer like this, as you can appreciate in this diagram. Now they are single cell thick layer. They basically they are the bone forming cells. So what they do is they deposit the organic matrix component, like they deposit collagen and the other proteins there. Now there are some cells, osteoblast cells, they are doing this secretory function, and they kind of come towards end of doing this secretory function, and whatever the matrix material they are secreting, no, they get trapped in that material only, and now they are inactive, they are not able to secrete the matrix, they are trapped, and these cells we call them as a osteocytes. So these are the osteocytes. So these are the resting bone cells, as they are not involved in any bone formation. They will present in the lacunae, as we already discussed, and they have processes which we call them canaliculi, and what they do is basically they maintain the integrity, right, of the bone matrix, as we already discussed earlier. And then now what happens, no, जैसे some आप लोगों ने कभी वो ऑब्जर्व किया है, जब कोई घर बनता है तो वो जो मिस्त्री होता है ना, वो क्या करता है, वो सीमेंट का ऐसे मारता है उस पर, और वो कब जब कम ज़्यादा जब होता है तो उसको ज़्यादा पार्ट निकाल भी देता है, राइट, टू मेक इट अ स्मूथ वन. So that kind of function is done by this osteoclast, isn't it? So bone surface has to be smooth. So yeah, remolding kind of function, that function is done by this osteoclast. So these are basically removing the unwanted material from the surface of the bone. So what they do, as you can see here, they lie on the, again the bone forming surface. They, as you can see here, they are the multinucleated cells, and basically bone resorption, bone remolding is their function. And on the other end, so this is the bone forming end where bone formation is taking place. On the opposite end, in this particular diagram you can see here, so there are some bone lining cells which are, they line the surface where like bone formation and resorption is not taking place, and basically transport of various nutrients and other substances, they are involved in that function. So these are various cells of the bones. So what we have seen is like osteoblasts, they synthesize collagen fibers, they synthesize matrix like all these proteins like osteocalcin, osteonectin, glycoproteins, all these are synthesized by this osteoblasts. And osteocytes ka function we already seen. What they do is they maintain the integrity, they maintain the form of the bone and help in the so basically a little bit help in the remodeling as well, but main remodeling ka function is done by osteoclasts. Yes, and we have seen that so there are some proteins and all, this matrix metalloproteins and all. Okay, so let us skip these points, it's not very important, you just go and read from your textbooks also. Yeah, so yeah, that's, yeah, so we have like dry bone, it's having 60% inorganic, then 35% organic components will be there. And what happens now, like when we can removal of these minerals which are there, like so they become so brittle, so soft that even we can tie the knot of them. We can do that in, and then if any bone is burned, so burning of the bone result in destruction of these organic components are there, and then they become very brittle, like as brittle like chalks. Okay, so that is what about the bones is. Yeah, so we'll stop here now, our time is up. So thank you very much. You please read from the textbooks. We have provided you the histologica manual. Read the points from histology manuals also. These are the key points, how to identify the slides in the lab. So when you go today for the practical, before you look into the slide, just go through those 10-15 lines which are given in the manual. That will help you in identification. Okay, read, discuss amongst yourself, and any doubt again you can come to me always. Yeah, so thank you very much. We will stop here. Yeah, thank you. Yeah. You may leave now. Yes, beta. Raja.